Today I'm going to talk about an important chapter, which is Principles of Inheritance and Variation. Before I proceed with further details of the chapter, you need to understand these two terms, this inheritance and variation. What do you mean by this? Inheritance means transmission of characters from one generation to next, that is from parents to offsprings. We see new organisms being born, maybe young ones of cat or young ones of dog, young ones of cattle, or even babies are born. So we see that these newly born young ones, or what we call offsprings, so this kind of resemblance of the organisms happens because the characters are passed on from previous generation, that is from parents to the offsprings. So this process of transmission of characters from one generation to next, this is what we call inheritance. Next, variation. The differences in characters of offsprings that we observe is called variations. So when you see these new organisms which are being born, you can see that each offspring, when you compare to the other, is not exactly same. There will be some variations. Even when you compare the offspring with the parents, there will be variations. So these kind of variations that we come across from one offspring to another, or when you come across one offspring, when you compare offspring to a parent, these kind of differences is what we call variations. The inheritance of characters and the variations follow a particular pattern. And Mendel, whom we refer to as father of genetics, has found that by conducting various experiments that this follows a particular pattern, and studying of this inheritance and variation helps us in a lot of ways. So what's the significance? What's the importance of this? We can understand the differences, like why some organisms are born as male, some organisms born as females. And we come across people with different blood groups. Some are A, B, AB, and O. So what's the reason for this? Inheritance and variation answers this. In humans, we come across lots of variations in variety of characters. Maybe it could be the skin color, or it could be the eye color. Some have brown, some have black. And in some people, the earlobes, it is attached or it could be free. In some people can roll the tongue, some cannot. So all these have the genetic basis, and principles of inheritance and variations answers this. And another major significance is about the genetic disorders. There are certain disorders like color blindness and some metabolic diseases like all genetic basis. And if we are able to understand this, we are able to predict what would happen, what could be the possibility of occurrence of this genetic disorder in the future generation, and we can look for possible remedies. So all these are the importance of studying this chapter. Another term that you need to know is homozygous and heterozygous. Homozygous means the one in which similar alleles are present, means alleles of same kind is called as homozygous. As you can see here, I have written TT, small t, capital R, R. So both alleles are of same kind. Then we call this as homozygous. Heterozygous means in an organism, there can be one allele of one type, another allele of another type. So two different kinds of alleles. So we can see in this that two different letters I have written, capital R, small r. So uppercase, lowercase. So if these two different alleles are present, representing whether it is uppercase and lowercase together, then we call them as heterozygous. So that means for any particular character, some organism may have same kind of alleles, whereas some other organism may have two different kinds of alleles. So likewise, we call them as homozygous and heterozygous. Another important term you have to understand is dominant and recessive. What do you mean by dominant and recessive character? When there are two dissimilar alleles, it is observed that the two alleles don't blend. Only one of the allele will express itself, whereas the other will be suppressed in the presence of the other allele. So accordingly, we call them as dominant and recessive respectively. So an allele that expresses its phenotype always means whether the allele, whether it is homozygous condition or heterozygous condition, this character will always appear. The phenotype will always be seen. Then we call this as a dominant character, and that the genotype that expresses is called as dominant allele. Same way, recessive allele, they do not express its phenotype always. That means when you look at a heterozygous condition, that is when there are dissimilar alleles, the expression of this recessive character is silenced. Only the dominant character will be expressed. But whether it will always remain silenced? No. Only when the dominant allele is not there, let's say in case of homozygous condition, then we can see the recessive allele expresses itself. So in short, I can say dominant allele or a character get expressed both in homozygous and heterozygous condition, whereas recessive allele will be expressed in homozygous condition only. For example, in pea plants, if you take, we say that tallness is a dominant character, whereas dwarfness is a recessive character. We can see that in a homozygous condition, TT, capital T, if you have written, the plant will always be tall. In a heterozygous condition, which has been represented as capital T, small t here, this plant will also be tall. Even though you have both alleles, one for tallness and one for dwarfness, you see that only tallness is expressed, but dwarfness is not. That's why it is dominant. You can see here, small t, we have written as a dwarf. So only in this homozygous condition, this dwarfness get expressed, whereas it is unable to express in its heterozygous condition. Another important term, monohybrid cross and dihybrid cross. Mendel performed monohybrid cross as well as dihybrid cross in his experiments. Monohybrid cross is when you are studying inheritance of a single character which differ in, of course, contrasting traits. Let's say you're studying only plant height, or you're studying inheritance of only seed color, or only seed shape, or only flower color. So you study any one particular character that which have contrasting traits. Then you call this as a monohybrid cross. So we say that a cross that involve plants that differ in one pair of contrasting traits. For example, seed color. You want to study only seed color. You're going to record the inheritance pattern of. So in a seed color contrasting traits, yellow plant, yellow seed producing plants and green seed producing plants. So these are the two contrasting traits, but you are studying only one character, that is seed color. Then you call this as a monohybrid cross. When you say dihybrid cross, it involves plants that differ in two pairs of contrasting traits. Means you study plant height with seed color, or seed color and seed shape, flower color, flower position. any two characters at a time. So totally you will be having two pairs of contrasting traits. Then we call this as a dihybrid cross. So a cross involving plants that differ in two pairs of contrasting traits. For example, here you're studying the inheritance of seed color and seed shape. Seed color have two traits, yellow and green. Seed shape have two traits, round and wrinkled. So you're studying two characters here. So it is a dihybrid. Single character is studied, that is monohybrid. Gregor Johann Mendel chose pea plants for his experiments on this inheritance and variation. Why this pea plant? Pea plant also we call scientific name is Pisum sativa. There are many reasons why Mendel chose pea plants for his experiments. Some prominent ones are these. Short generation time. So for any genetic experiments, if somebody chooses an organism which having a long lifespan, that would be not a better idea, because you will not be able to get many generations to compare how many characters are inherited and how many are there is a variation. So having a short generation time is very important when choosing an organism for any genetic experiment. So a pea plant is an annual plant. It completes its life cycle within a year. So you can obtain so many generations for a period of time, and you have data to derive. And you'll be able to, based on the data, you'll be able to say what exactly and how many inheritance variations have been there. So short generation time is one of an important feature. Apart from this, variations in numerous characters. We can see that in a pea plant population, we see variation with reference to so many things, like there is variations observed in stem height. Some pea plants are tall, some are dwarf. We can see variation in the flower color. Some are violet, some are white. Seed color variation, yellow, green, and seed shape, round, wrinkled, flower position. So we can see numerous variation in pea plant population. We don't see such kind of variations being observed in many other plants. So when you have so many variations, that will be an ideal plant for experiments. It can be self-pollinated and cross-pollinated. Self-pollination, if we want to complete, do a genetic experiment with the same genotype, Mendel actually self-pollinated. If you want to try with plants of different characters, then you may have to take the pollen of one plant, put it on the stigma of the other plant. So there is a cross-pollination. So this, not all plants allow both self-pollination and cross-pollination. So whereas pea plants can be self-pollinated and cross-pollinated, and we can obtain the offsprings, we can raise the offsprings and see the results. So all these are the advantages of choosing pea plants as a model organism for genetic experiments. Mendel, when he was working with pea plants, he focused and concentrated his studies on 7 characters of pea plants. So which are those 7 characters he studied? So those are stem height, seed color, seed shape, pod color, pod shape, flower color, and flower position. These 7 characters. So we know that each character had their contrasting traits. Stem height, tall, dwarf. Seed color, some pea plants were yellow, some were green. Seed shape, round, wrinkled. Pod color. Pod is nothing but it's the fruit that bears the seed. Its color green, which is a dominant character, and yellow is the recessive trait. Pod shape, inflated. and constricted is the recessive one. Flower color, violet was dominant trait, whereas white is the recessive trait. These were variations. Flower position, axial and terminal, means the flowers are seen only at the tips. Those are the terminal. Axial all along the stem and branches you come across. So then we call it as axial. These were the characters and their dominant and recessive traits that Mendel studied for his experiments. Mendel conducted this monohybrid cross experiment. In a monohybrid cross experiment, inheritance of only one character that differ in contrasting traits is being studied. Here we are studying, taking one example, that is stem height as an example. So out of 7 characters, any one we are chosen, stem height here. Any other example, if you choose, you get the same results. Now, in stem height, so Mendel chose for his starting experiment what we call parental generation. He chose tall plants and dwarf plants. Tall plants, homozygous tall plants, both alleles are same. Again, dwarf plant, they are obviously homozygous. Now, when he crossed these plants, you see that this plant produced the gamete, all will be of same kind, capital T we are representing. This produce the gametes, all will be of this kind, small t. So when the gametes fuse, you get the plant which have dissimilar alleles. So this is what a heterozygous. But we see that only one trait is being expressed, that is T is expressed, but not dwarfness. So that's why they have written it as tall. So tallness is expressed because it is dominant, and dwarfness is recessive and it is not expressed in a heterozygous condition. So when you obtain the plants, offsprings from this generation, all the offsprings were tall because of this reason. Then Mendel self-pollinated the F1 generation. This is what we call F1 generation, or the first filial generation. Now he self-pollinated. So which is represented like this. So self-pollinated. Now here, as they do have dissimilar alleles, you can see that this organism is capable of producing 2 types of gamete. One is capital T, small t. This also is capable of 2 kinds of gametes. Now, when these 2 gametes are produced, what are the different types of offsprings that we get? That can be easily predicted by using Punnett square. So this T small t here, we write here, and capital T small t, we represent like this. And you see that capital T here, capital T small t, capital T small t, small t small t here. So these are the kinds of offsprings that we get, which I have written here. If you look at the genotype here, so out of these 4 offsprings which has been shown in the Punnett square, so we get homozygous tall 1, heterozygous tall. 2 of them will be heterozygous, 1 will be again homozygous dwarf 1. So the genotype that we get is 1:2:1. But what is their morphological feature? What is their external feature? We can see that this will also be tall. These 2 also will be tall. So there will be 3 organisms which will be tall and 1 will be dwarf. So the ratio of Mendel's monohybrid cross is 3:1. So which is the phenotypic ratio. The genotypic ratio is 1:2:1, where you specifically look into the genotype of each. Otherwise, the ratio, or when we say Mendel's monohybrid ratio, it is 3:1, which is nothing but the phenotypic ratio. So if we take any character out of those 7 which is studied, he obtained the same ratio, that is 3:1. So if a Mendel in this generation, that is represented as F2 for second filial generation, if he has got 100 plants, about 75 plants will be of a dominant character. That means it resembles the dominant parent. And 25 plants out of those 100 would resemble the recessive parent. That means those plants would be dwarf. So 75% would express a dominant character and 25 would express a recessive character. So this is 3:1, which is Mendel's monohybrid cross ratio. This holds good for almost all cases except few exceptions which were discovered later after Mendel's time. Based on Mendel's monohybrid cross experiments, Mendel was able to derive the laws. Which are those two important laws? One is law of dominance and law of segregation. This is the Mendel's monohybrid cross that I represented as I've explained in the previous slide. So what is this law of dominance? Law of dominance states that when a pair of factors, Mendel called them as factors, P and small, when a pair of factors controlling contrasting traits, that is tallness and dwarfness, the other two contrasting traits. When a pair of factors controlling contrasting traits come together in a F1 hybrid. So this is the F1 hybrid, and they come together in a F1 hybrid. Only one of the factor will express. In this case, let's say capital T will express. The other factor is not expressed. The one factor which is expressed is called dominant. The other factor is recessive. This is what we call law of dominance. So law of dominance says that when a pair of factors controlling contrasting traits come together in a F1 hybrid, one of the factor which we call dominant is expressed and the other is not expressed. And this is called as law of dominance. Then what is law of segregation? Law of segregation states that when a pair of factors controlling contrasting traits, that is tall and dwarf, come together in a F1 hybrid, they remain unmixed. When we say unmixed, it means it doesn't show any blending. If they are present, but it remains unmixed. But later, during gamete formation, they segregate. You can see that the organism has both, but it has present in that organism, but it has not undergone any kind of blending. But it is capable of separating, or what we call segregating. When does that segregation happen? It happens during gamete formation. That's what we see. Because of the segregation, we can see that these are the gametes segregated. That's why we are able to get the dwarf plants once again in the F2 generation, which was disappeared in the F1 generation. So the law of segregation states that when a pair of factors controlling contrasting traits come together in a F1 hybrid, they remain unmixed. That is, they do not undergo blending. But later, they are going to separate. When does it happen? They are going to separate during gamete formation. They are going to segregate during gamete formation. So this is called as law of segregation. Mendel's laws hold good in most of the cases, but there are some situations, some examples have been found where Mendel's law is not applicable. So these are what we call deviations from Mendel's law. So here I'm discussing an example where Mendel's law is not applicable. Mendel's law of dominance is not applicable in this situation. This is observed in case of flower color in snapdragon, or scientific name Antirrhinum majus, or in flower color in case of 4 o'clock plant, or we call it as Mirabilis jalapa. So in this plant, we observe red color flowers, white color flowers, but also pink color flowers. So as Mendel had said, there is no blending in F1 generation, but here we come across a blended appearance. So we can see that red flowers we represent like this with homozygous condition, and white flowers this condition. And in F1 generation, we got heterozygous, but here red color is not seen. So one allele is not able to completely dominate over the other. So this is what we call incomplete dominance. If there is a complete dominance, as I had explained by Mendel, it would have been red color, but there is an incomplete dominance. So we get an intermediate phenotype, which is pink color. Again, if we continue with this, and in F2 generation, we get 1:2:1 ratio. That means whatever the genotype is, in the phenotypic ratio is also the same. Whereas in regular Mendel's monohybrid cross, we used to go to 3:1. Whereas here 1:2:1, because the heterozygous condition will have a different phenotype compared to the dominant parent. So 1:2:1. So 1 red, 2 pink, and 1 white. So thus, this is a deviations from Mendel's law of dominance. Like incomplete dominance, there is also another situation where Mendel's law of dominance doesn't hold good. This situation is codominance condition. According to Mendel's, one of the allele will dominate over the other, so that one will express, the other one will not express. What if both of them do express? What if both of the alleles start expressing without one dominating over the other and without other alleles getting suppressed? So when both of them do express, then we call this as codominance. This is seen in AB blood group type. So I can show this with an example. An individual with A blood group, homozygous A, another individual with B blood group, homozygous B, and when gamete is produced, IA and IB. So the F1 individual will be IA-IB. This person will have AB blood group. So here, neither A is dominating over B, nor B is dominating over A. Both alleles are expressing. So the person will have AB blood group, so which is different from this parent or the other parent. This is because of codominance. Again, in F2 generation, we observe the ratio A blood group, AB and B 1:2:1. So as the heterozygous condition will have a different phenotype, you will have 2 here. So 1:2:1. The regular Mendel's monohybrid cross ratio was 3:1. Here we come across a deviation 1:2:1. So thus, both in case of incomplete dominance and codominance, the Mendel's monohybrid cross ratio will not hold good. Here, both genotypic ratio and phenotypic ratio will be the same, which is 1:2:1.